So thank you all for joining us. It gives me great pleasure to introduce Professor Fale, the academic dean of our college. As with all things with Professor Fale, his presentation is, I think, very mildly related to dentistry. It's very broad, and as he said, this research had got the Nobel Prize for Medicine and Physiology in 2021. So it will be an unusual presentation that we usually as dentists don't get to hear, and only someone like Professor Fale can give that to us. So with great pleasure, I invite Professor Fale.
Thank you very much, Dr. Nebo. So they told me that because it's a CPD presentation, I need to have objectives. So these are my objectives for the talk. I'm going to talk about ice cream, garlic, cinnamon, mustard, and menthol. Okay. This presentation is about why sometimes we feel pain when we eat ice cream in our teeth, why sometimes raw garlic tastes different than cooked garlic, okay? And some researchers that have actually, this thing we all know, right? How many of you guys have noticed that, you know, the raw garlic, it's a bit, you know, it's a bit, sometimes when you put in your tongue, you feel it there, right? It's raw garlic. Whereas cooked garlic, it doesn't, right? It's something that everyone, has anyone here not eaten garlic sometimes in their lives? No, right? Well, so there is some scientists that actually looked into that in depth, okay, to try to figure out, to get to the bottom line, why you feel something funny in your mouth when you eat garlic, why you feel pain in your teeth when you eat ice cream, okay? And by looking deep into those things that seem so banal and so simple, okay, but when you look into something like that so in depth, they actually made huge discoveries and ended up winning the Nobel Prize. That medal over there is the medal of the Nobel Prize. So the reason I wanted to give this talk is for two main reasons. One is the garlic, the other one is the ice cream. No kidding. One is the Nobel Prize in Medicine that was given in 2021. And the other one is a very important paper that was published in Science Advances last year as well, that they're all both related. So actually in my presentation, I'm going to talk about those two papers. But the first thing I wanted to talk is about the person that won the Nobel Prize this year. So his name is Artem Patoputian. So he's Armenian but Lebanese. He's Lebanese Armenian. He was born in Beirut, and he went to primary school in Beirut. He went to high school in Beirut. Okay, and then he did 1 year of university in the American University of Beirut. But because of the civil war in Beirut, his family moved to the US, and then he continued his studies in the US. And he became a very renowned scientist in the field of neurophysiology. And for his discoveries on the receptors of pain and temperature, he was awarded the Nobel Prize last year. Okay, so there is some amazing people coming out from the Middle East that can achieve great things. The other reason I wanted to talk about him and his research is the NIDCR. So for those of you that are not familiar with this thing, the NIH is the National Institute of Health, is the #1 institution of health research in the world. Okay, it's based in the US. Okay, they fund research in many fields related to healthcare, and it has a particular institute that is related to dental research. It's called the NIDCR, National Institute of Dental and Craniofacial Research. It was established in the 1920s, 1930s in the US, and it has been spearheading dental research in the US, and has many of the discoveries that we know that are related to dentistry were funded by NIDCR. So this particular Nobel Prize, this gentleman from Beirut that won the Nobel Prize, he was funded by NIDCR. So this research that won the Nobel Prize was funded by a dental research institute. Okay, and there's not many of those in history. That's another reason I wanted to talk about this case. Even though I'm not a neurophysiologist, I have very little knowledge about neuroscience or receptors, but I thought it was an important thing to share with you guys this kind of important things that happened last year. Okay, so the main discovery of Dr. Patoupyan that got the Nobel Prize for is a particular molecule that is on the cell, on the membrane of some somatic nerve cells, okay, some neurons, is the TRPA1. Okay, this is a channel that is on the membrane of the cell that allows the movement of calcium, okay, when it gets activated, and it's sensitive to something they call a scalding temperature, like the feeling of that you're getting burned, okay, and it's also sensitive to some chemicals. Okay, so it has many implications in healthcare, you know, and for many things. Okay, when you put your hand in hot water and you feel this sharp pain that you want to take your hand out, it's this receptor. So he discovered that. Okay, but he also looked into that receptor in the mouth, because it turns out that it's very abundant in the mouth, especially in the tongue. Okay, so we as dentists, we work with the tongue every day. So in the tongue, there is some very sophisticated biochemistry happening, okay, that usually as dentists we ignore completely. We only care about drilling holes in teeth. we don't think further than that. Okay, so he published many papers about this receptor and all its mechanisms, and based on all those papers and all his findings on this particular molecule, he was awarded the Nobel Prize. But I'm gonna focus on one particular paper that I found that was particularly interesting. He published it in 2005. That was specifically about the mouth, okay, which is a paper in which he looked at this receptor in the tongue, okay, and its role in how do you taste garlic, okay. He published this paper in a journal that is not one of the top, but it's pretty high. It's called Current Biology, so it has an impact factor of 10. Okay, so just to give you an idea, dental journals, like the top dental journal is around 7, Journal of Clinical Periodontology. This is a bit higher. Okay, so garlic is used all over the world for cooking and many things. People use it also because they think that it has medicinal properties. And garlic has a funny taste, in particular because it has some sulfur molecules in it. And everyone knows that cooked garlic and raw garlic tastes different. Okay, and there was some research before they did the study that they found that when you put raw garlic in the mouth of a mouse, you get some activation in some particular nociceptors neurons in the trigeminal ganglia. Okay, the trigeminal ganglia is the one that regulates sensation of the oral cavity, right? The sensation goes to the trigeminal ganglia, so it gets activated when you put garlic on the tongue. That's why when you put garlic on your tongue, sometimes you feel a bit funny. Okay, so in this particular study, he showed that raw garlic in particular activates this particular receptor, the TRPA1 and the TRPV1. Okay, they are temperature-related receptors. So that's why when you get the garlic, you get this, you know, this feeling, you know, that is a bit like burning. And they also found which molecule in the garlic actually activates it. Okay, so there is a molecule in garlic that is called Allicin, and this is the molecule that indeed activates these receptors in the tongue and make you feel the funny feeling when you eat garlic. Okay, so before they did that study, they already had done other studies on these families of receptors in the tongue. So they discovered, for example, that the same receptor, the TRPV1, is the responsible for, you know, you feel a burning sensation in your mouth when you eat chili. Okay, so the chili has a molecule that is called capsaicin. Okay, and that molecule interacts with the receptor in your tongue. Okay, and gives you the burning sensation. This family, there is another group on this family of receptors, which is the TRPA1. It's also related to temperature and pain, but this one is activated by mustard and by, for example, the cinnamon. Okay, that's why when you eat cinnamon, also you get a funny feeling in your mouth, also with mustard. Okay, and in the same class of receptors, you have the TRPM8, and this one has to do with the feeling of coolness. Okay, it's a receptor for cold temperature. So you have it in your mouth, and that's the reason why when you eat a menthol or a chewing gum with menthol, you feel that your mouth is cold. Also, when you brush your teeth with a toothpaste that has menthol, you feel some coolness in your mouth. It doesn't mean that your mouth got cold. It's just that it's just a trick. Okay, your mouth is the same temperature. It doesn't get cold. It's actually, but the feeling, you get a feeling that it's cold because this receptor got activated by menthol. Okay, these are also some of the discoveries made by the group of Dr. Patonian. Okay, so many cool stuff happened in the mouth that ended up in a Nobel Prize. So they knew about these receptors, and they wanted to find out if how garlic fits in these receptors. So they did some research to find out which one of these receptors gets activated by garlic. Okay, so over here there is this is a panel of their paper in current biology. So in high impact journals, usually they don't have figures, they have panels of figures. Okay, so this is figure number 1. Okay, and you can see how many small figures they have, because they collect a lot of data because they want to make sure that they got their science right. Okay, so they do lots of experiments just to confirm from multiple angles their findings. Okay, so this figure number 1 in the paper over here, figure 1A, they're doing an experiment in which they have the receptors, okay, and they're looking, these receptors, when they get activated, the temperature receptors, the different types, they increase the concentration of calcium inside the cell. So they have a special technique in which they can measure the amount of calcium inside the cells when they get activated, when the different receptors get activated. So they are adding extracts of garlic, okay, to the cells, and then they see the signal. So for example, here in green, and they look at the signal when they added baked garlic, and there is no signal. Okay, there was no calcium going to the cell didn't get activated. Baked garlic, cooked garlic doesn't really activate these receptors, but when they added fresh garlic, then they get a signal. Okay, and they get a signal in 2 receptors, in TRPV1 and in TRPA1. Both of them, they get activated by the fresh garlic, but not by the baked garlic. Then over here, in this graph over here, they're doing another experiment, which is an electrophysiology experiment. So here, they're looking at the electrical current in a nerve, okay, when it gets exposed to garlic, for example. So they had the receptor, the TRPV1 receptor activated by garlic, and they got an electrical pulse. So the neuron responded, okay? And then they did the same thing with the other receptor, and also they got a response. So they saw in the cells that the calcium was increased in the cells when they got exposed to garlic, and it was activated by this receptor. And they also looked at the electrical impulse to the nerve when they put the garlic on this receptor. So then in 2 different ways, they are demonstrated that, yeah, this receptor in the cell is activated by garlic. Here, they repeated the same experiment in cells from different animals. Okay, so they were not okay just to try with the mouse. They tried with cells coming from the mouse, from the rat, and from human, and they got the same results in all of them. Okay, so they wanted to make sure that all the findings are consistent. So they could say that, this receptor is responsible for the garlic feeling, and you see it in mice, in humans, and in rats. Okay, here is another experiment in which they looked at a particular molecule in garlic that is called allicin, and it repeated the same experiment, but instead of giving garlic to the cells, they give allicin. Okay, and they found the same thing, like the signal, the receptors get activated by allicin. The calcium concentration in the cells increases when you put allicin on the receptors, and this happens with cells from humans, rats, mouse. They look at the electrical current in the nerves when they add allicin, and also they get an electrical pulse. Okay, so this is to show that In garlic, this particular molecule, allicin, is the one responsible for activating the receptors and for giving this funny feeling when you eat garlic. Okay, this is an experiment in which they look at the activation of the cells when they add different compounds. Okay, so they have the cell and they had they added the allicin, the garlic, and also they added the capsaicin, which is the one of the chili. Okay, so they add allicin, which is the molecule that they found in garlic itself, and the chili. All of them activate the cells. And this is regarding one of the two receptors. This is the TRPV1. Okay, in the TRPV2, the allicin, sorry, the so this is the TRPA1 and TRPV1, and this is the TRPV1. Here they get very strong signal with allicin and with the chili. Okay, so the allicin and the chili, they activate these receptors. And this is another experiment that they did to show the difference between baked garlic and raw garlic. So what they use is they use nuclear magnetic resonance machines. Okay, so NMR machines. So these are the same machines that used to make images for patients in the hospital, like NMR machine. You can use it also for chemical analysis. Okay, so NMR machine, actually they were invented for chemical analysis. So they put in this machine extract of garlic, and this machine gives them a signal. Okay, and this is the signal that they get. Okay, with different peaks that correspond to the different molecules that you can find in the garlic. And then they put they cook the garlic and they put it back in the machine. And the signals are different. Some molecules are there, but some molecules have disappeared. Then they put allicin in. Allicin, the molecule that they think that it's responsible for the feeling in garlic. And the allicin, when they put the machine, it has these peaks over here. Okay, and you can see that these peaks were also present in the fresh garlic, but when they bake the garlic, they disappear. Okay, so this molecule is present in garlic when it's fresh, but is absent in the baked garlic. So the cooking of the garlic destroys this molecule and disappears. Okay, and that's how they can show that, okay, this molecule is activating the receptor in the tongue, and that's how when you eat the garlic, you feel this funny sensation. Okay, so they had a conclusion in the article. Very simple. Okay, after huge amount of work. Okay, it's just one sentence. In raw garlic, allicin is the active ingredient that causes a burning sensation. Okay, through activation of TRPA1 and TRPV1. Okay, so this is a typical conclusion that you will find in a very high-end biology paper, because it has two parts. One is observation. Okay, allicin is the active ingredient that causes burning feeling in the mouth. Okay, and that will be enough for many researchers around the world just to show the observation. But they go one step beyond and they talk about the mechanism. Okay, and say, oh, this is the molecule that is causing the burning sensation, and it's because of this. Okay, so when you're doing really good research in biology, particularly, you should have both things: the observation and the mechanism. Okay, you cannot never explain the why, but you can explain the how it happens. So they are explaining the how. Okay, if you want to submit a paper to Nature or Science in anything related to biology, if you don't have the how, there is no way to go through. You want to see the observation and the mechanism. So that's the paper from 2005 from the team that won the Nobel Prize. But following on this same area of research, last year there was also a very important paper that was published in Science Advances, and it's also dental related, and is building on the previous discoveries of the team that won the Nobel Prize. So Science Advances is one of the most prestigious journals in the world. It has an impact factor of 14, and this article is very, very relevant to dentistry because it's actually done in the tooth itself. Okay, and one of their main findings is to explain why your teeth hurt when you eat ice cream. Okay, but it has other implications related to caries and tooth sensitivity. Okay, so it's very well known that when you eat something cold, you can feel a sharp pain in your teeth. Also, people that have inflamed teeth because of caries or pulp irritation, their teeth are very sensitive. Okay. It's also well known that the enamel acts more or less like an insulator for the tooth that protects from the environment. Okay, and then when you have the irritation in the pulp, the irritation in the tooth or caries, the dentin can get exposed, and then you have this irritation. And for many, many years, it was believed that the pain and the sensitivity that you get in the tooth, it has to do with the exposure of the dentinal microcanals. Okay, the dentinal microcanals are exposed to the environment because of the caries or because of recession or for whatever reason. And there is movement of fluids inside the canals. And this movement of fluids is the one that is actually causing the pain. So that's the hypothesis, the theory that has been going for decades in dentistry. That's what we learn in school, dental school, forever, until last year, because last year this team of researchers demonstrated that this theory is wrong. Okay. Okay, so in 2022, this is not the right explanation anymore, because they discovered that there is an actual receptor in neural cells in the tooth that are very sensitive to cold, and this is a receptor that is causing this sensitivity of the tooth. Okay, so they were looking at different potential explanations for the sensitivity of the tooth. They looked into different receptors in the family of those that I explained earlier that were awarded the Nobel Prize. They are called the TRP receptors, the transient receptor potential ion channel receptors. So they looked at, for example, TRPM8. Remember, this is the one of the menthol, okay, that gets activated when you eat menthol or with the toothpaste. TRPA1, this is the one of the garlic that gives you the stinky sensation with the garlic, okay. They found that these ones get overexpressed in the ganglion, the trigeminal ganglia, when there is a irritation of the tooth, okay. But the expression is not that high, okay, and also some of these receptors were not found in odontoblasts, for example. Okay, so they decided to look into this new receptor. It's called TRPC5. Okay, so the previous ones were the one of the menthol, the one of the chili, the one of the garlic. Okay, now they're looking into a different one. It's called TRPC5, and the reason why they looked into it is because they discovered that in the trigeminal ganglia, when they analyzed the RNA in the trigeminal ganglia, they found that there was mRNA for this receptor. So apparently, the trigeminal ganglia, the cells, the neurons in the trigeminal ganglia are producing the proteins of this receptor. So that's why they decided to study it. And it was known to be involved in cold sensation in the skin, but no one has looked at it into the tooth. Okay, so this is the receptor. It's a calcium channel kind of receptor that wasn't really understood until last year. Okay, so people have associated it with mercury toxicity, with some neurological behaviors, but last year was the first time it was found that it had a role in the sensitivity of the tooth. So the first experiment this group did, it's a very cool experiment. They brought some mice, okay, and they create an irritation in the tooth of the mouse, okay? And for some reason, when you create an irritation in the tooth of the mouse likes to, it's completely counterintuitive, the mouse likes to drink water with sugar, okay? They seem that they have some suicidal behavior, the mice. And for some reason, the mouse, when they increase the sugar consumption, when they have an irritation of the tooth. Basically, the opposite that they should be doing, just like children. Okay, so they have 4 groups of mice. So one group of mice is just a regular group of mouse. Okay, just they call them C57BL. So these are that's their name of this type of mice. They are basically normal mice. But then they have mice that are genetically modified. Okay, so these ones over here, they represent mice that are genetically modified to lose the receptor of the menthol. Okay, and they are genetically modified to have absent the receptor of the chili and the garlic. Okay, and there is a group of mice that didn't have the receptor of TRPC5, this new receptor that they were investigating. So they looked at the consumption of sucrose by the mice before doing the dental irritation and after. Okay, so this before and this after. So in a regular group of mice, they are drinking some sugar before, but after they drink 3 times more. Okay, and the mouse that didn't have the receptor of the menthol, the same thing. You know, they were drinking some sugar before. After they got the pulp irritation, they drink more. Then the mouse that had the chili receptor blocked or eliminated, the same thing. But then in the mouse that have the TRCP5 receptor eliminated, they didn't feel anything. Okay, the behavior before and after the irritation to the pulp was the same. So that was the first hint that maybe that receptor is playing a role in the sensitivity of the tooth. So then they went on to do a more sophisticated experiment. It's actually a very, very sophisticated experiment. They took a mouse, okay, they sacrificed the mouse, the poor mouse, okay, and they took out the mandible. And they took out the mandible with the nerve, okay? And they put it in an incubator, okay, with fluids there to keep it alive, keep the cells alive. And they plugged in the nerve to electrical machine that could detect electrical signals. Okay, so that way they could play with the dentin, okay, and the enamel, and then see what signal they get on the other end. Okay, so they can do different tests. Okay, and they could compare, for example, with the skin. So one thing they noticed, one of the first experiments they found is that the sensitivity of the tooth for cold temperature when they did this experiment was much higher than the sensitivity of the skin for cold temperature. Okay, so this is they expose the tooth for cold temperature, they expose the skin for cold temperature, they measure the nerves, and these are the results of the activation of the nerves. Okay, so you can see that the tooth, the activation was higher than the skin. Okay, and it's kind of normal. When you put ice on your skin, yeah, it's annoying, but it's not that bad. But you put ice on your tooth and it's gonna hurt, right? Like I'm sure my endodontist friends, they can confirm that. Like you can put what do you use for testing the pulp vitality? What do you call it? Endo ice. If you put it on your skin, does it hurt? Not really, right? But it hurts. So basically that's what the endodontists know already for many years. That's what they show in this experiment. Okay, so they confirm that. The tooth is very sensitive to cold, and they were able to measure it in the lab. So then once they have a well-established model to test that, then they went forward and start to test the hypothesis whether or not this receptor is actually the one responsible for the pain sensitivity in the tooth. So they did many experiments. I'm going to try, this is a panel that is very crowded. I'll try to go through it in as simple as possible. So let's focus on this part over here first. Okay, so here they have a group of mice, okay, in which they are changing the temperature. Okay, so over here in this part of the graph, you have the temperature. So it goes from room temperature to cold and then back. Okay, so from room temperature to cold and then back. And over here you have the signal, the pain signals that they get in the nerves. So you can see that there is no pain signals here, but then when they cool down the tooth, you get pain signal. And then when it warms up back again, the pain signal goes. Okay, so there is a lot of pain signal when it's cold. When it's warm, it goes away. They do the same thing again, the same thing happens. Okay, cold, sorry, hot, cold, hot. Okay, and then they get the signal when it's cold. And then they add a molecule into the solution, a molecule that is similar to menthol. Okay, similar to menthol, so it blocks one of the receptors related to cold feeling, and then the signal goes a little bit less. Okay, but there is some signal there, and then they they repeat the same thing again, and they add a molecule that is specifically targeting the receptor they're looking into, the TRPV1, okay, and then the signal disappears completely. Okay, so it could be a very interesting spice, you know, to put in the food, this this new molecule, something that you could put in toothpaste for sensitivity teeth, okay, because it blocks completely the signal. So these teeth are not feeling pain anymore. Okay, then they do the same experiment this is the same experiment, but in a different type of mice. These are in mice that don't have the receptor. Okay, so these are mice that don't have the receptor. So in these mice, it doesn't matter if you're cooling or heating or cooling or heating or cooling the tooth doesn't feel anything. Okay, if they don't have this receptor, okay, the tooth doesn't feel anything. So that's one way of proving their hypothesis that this receptor is really important, just by testing on a mouse that doesn't have the receptor. Okay, and these are very common practice in molecular biology research and in biological research. You know, when you want to know what's the function of something, you take it out. Okay, when you want to know what's the function of this receptor, you take it out and see what happens. Okay, so that's what they did. Okay, these are subsequent experiments in which they look at the electrical signals in the nerves of different types of mouse and mice that don't have this receptor at all. So here is the healthy mouse with without the receptor for the menthol, mouse with partial removal of this particular receptor, the cold sensitivity, and mouse that doesn't have it at all. So the mouse that doesn't have it at all, the nerve doesn't respond. Basically, it's always like that, you know, that is not responding. So in multiple experiments, different angles, they're confirming the same thing again and again, that this receptor, when you eliminate it, there is no feeling of pain due to cold. Okay, so that's the second panel. Okay, the third panel of the study, in which they show that this receptor is important there. The next panel is one of my favorite ones because it's very beautiful. So this one is a very complicated experiment. Okay, in this one, what they have done is that they have modified genetically a mouse. Okay, so that in whatever place in the body they are producing this receptor, and their UV light is going to glow with the green color. Okay, so any spot in the body where they're producing the receptor is going to glow in green color. Okay, and then they have another molecule which is the beta B3 tubulin, which is like control. It will go in red. Okay, but the receptor will go in green. The way they do it is that they get the DNA of the mouse. Okay, they look at the gene that is specific of TRPC5, and they bind that gene with another gene from a squid. Okay, and the gene from the squid is a gene that the squids use to make fluorescent molecules, okay, of different colors. Okay, so that means that whenever the mouse is going to produce this receptor, by default, it's going to produce the molecule of the squid. Okay, so you have a mouse squid. Okay, so that means that in any site of the body this receptor is produced, it's going to have a green color and the fluorescent light. So that way, when they got the tooth of the mouse, they dissected the tooth of the mouse, they put it under UV light, and then boom, you get the receptor glowing in green. So you can see how abundant is the receptor in the roots of the tooth. It's everywhere. Okay, it's all over the dentin. It's in the dentinal tubules. Okay, it's small branches of the nerves getting into the dentinal tubules. Okay, so these molecules produced by nerves, not by odontoblasts, and it's inside the dentinal tubules. Okay, and this is the last slide in the study. That okay, very nice. They found all these things in the mouse. So what about humans? So they got a few human teeth that have caries and no caries teeth, and they try to look for the receptor inside the tooth. Okay, so here in this case they use immunolabeling. So they use some antibodies against the receptor, okay, to stain it in the tooth. Okay, so in this case the receptor is going to be colored in red. Okay, so this is the tooth, and here these are different areas of the tooth that they looked into. Okay, that are magnified over here. So this is B is here, C is here, and D is here, okay, the high magnification. And you can see in different spots of the tooth, you can see the red lines, okay, the red lines that are related to the TRPC5 receptor. So they can see that this receptor is present in the dentin in many places in the tooth, okay. And then the other thing they found, they looked into is in a tooth that has caries, okay, that has some pulp irritation. So here they also found it a lot, and they even found it more abundant than in the healthy tooth. Okay, so they did quantification of the amounts of the TRCP5 receptor. So this is in a normal tooth in the root, and a tooth for pulpitis. So you can see the tooth with pulpitis is much, much higher. Okay, and they do the same thing in the pulp, normal pulp with pulpitis. It's much, much higher. Okay, so it seems that when you have an irritation in the tooth, the nerves in the tooth start to produce this molecule, and they make the tooth more sensitive. Okay, that's why your tooth becomes more and more sensitive when you have a pulpitis or a caries. Okay, so this is the final conclusion of the study, that TRPC5 is the cold sensor healthy teeth. Okay, TRPA1 it combines with the other one and gives the full cold sensation. So odontoblasts seem to have this receptor, okay, and they play a role in the cold transduction, okay, and provides a mechanism for prolonged cold sensing via TRCP5. Okay, so basically the reason why you feel cold and pain in your teeth is not the movement of water in your dentinal tubules, but is the activation of the receptor in the odontoblast and the nerves of your pulp. Okay, so as a summary, this family of receptors, they call them thermo-TRPs. They are class of temperature receptors that you have in your mouth and your teeth. They play a key role, feeling the cold when you eat the ice cream, feeling some nice coldness when you brush your teeth with a menthol containing toothpaste. That's the reason why you feel spicy food in your mouth when you eat chili or mustard or garlic or cinnamon. And they told me that because this is a CPD credit, I need to put this slide on at the end. If anyone wants the credits, I think they need to contact probably Dr. Ala or Dr. Nebu. I guess you said, okay. And that's it. And thank you very much. I apologize for the too scientific talk.
You're open to questions now?
Can I ask a question? So, Fale, does that mean that our traditional teaching on showing those odontoblasts and movement of fluid and the nuclei going up, shall we discontinue that now and remake those slides?
Well, one of the funny things that I learned reading the paper on Science Advances was that they, particularly in the introduction, they are discussing that issue. And they say, okay, this is what has been taught for many years, this is what the assumption has been, but the reality is that there was no evidence about it. It was a theory. So they went back in the literature to look for the evidence, and it was just a theory. It was never demonstrated. So the answer, I think yes, maybe we should consider revisiting those slides and update the knowledge. Yeah?
So does this reject all other theories, or maybe just explain?
No, that particular one, the sensitivity.
Could it be a combination of both? I mean, maybe we can work on the older theory and try to also prove it, and maybe it's a combination of both theories.
Well, the thing is that the other one hasn't been proven. So if someone does experiment to prove it, that would be great, but it hasn't been proven. Yeah. Like there's many examples in dentistry of things that we assume as facts, but because people have repeated it many times, but when you go back in time, where does it come from? It was just sometimes opinions, and this is another example.
Because the theory of the fluid might not be totally wrong for other types of pain or sensation that the tooth feels or the person feels through it. So it is like not a total rejection, rather alteration of.
Like the bottom line is that maybe the fluid and the dryness when you dry the tooth or whatever, it's cooling out the tooth, and the pain is caused because this receptor is activated when you maybe
Yes.
dry the tooth or whatever, you know. But the bottom line is that this is a receptor that is causing the pain.
In a way, because the sensation of a tooth depends on three theories. It's not only one theory. This is number one. And now, the first part of the paper, it talks about the cold sensation. Then what about the hot sensation, for example? What about the electric sensation? What about the mechanical sensation?
I think the thing is that you will find that will also be published soon, because now generally since the all that work on the thermal TRPs or whatever was done, now the general view is that almost all the sensation, pain, sensitive, it is through expression of certain molecules. But a lot of those molecules have not yet been found. But eventually they will be found.
I think it's a field of research that is very promising, like the neuroscience of the tooth.
I mean, I personally never trusted any of the theories. I read it, I taught them, but I never trusted. So this is very interesting. But perhaps you know what the comments have been made. Expression of TRPV1 receptors and those molecules may be one of the mechanisms, but surely it doesn't rule out other possibilities. So this is established now, but then there may be more to it than we know at this stage. So my question really is, since this research has been published, has it actually translated in, has this been incorporated in any of the strategies for managing dentine sensitivity?
That I don't know. I don't know, but I think that will be something very interesting to look into.
Because this is going to translate into billions of dollars.
Yes. Yeah, I'm
So
sure
this is nothing minor.
They'll have to find something to suppress the expression of this molecule, and that would
Well,
cure
they have it.
sensitivity.
In the paper, they have a molecule that blocks the receptor. So I'm sure they might have patented it or something. They have has a name that is many numbers and letters.
So whosoever gets the patent,
Yeah.
there's many
they can sell it
billions
for
of
good.
dollars to make.
Yeah, you see, almost everything which we have learned comes down to these things. I remember, I will actually find the paper and give it to Nabu to share with you. Amelogenesis imperfecta. Again, it's, all the theories on amelogenesis, this and that, it's all down to expression of certain molecules. And in Leeds, they did a study where they found a drug which is already used in children who have a particular type of disease. I can't remember which one. And they hypothesized that because that drug suppresses that particular thing in those children, it should suppress amelogenesis, expression of amelogenesis. And they did the study with an amelogenesis gene in mice, and amelogenesis did not occur in those mice. Because that particular, I will find that paper. So I think almost everything now comes down to expression of things and suppression of that expression to make things better.
As a clinician, I was more concerned within the management of dentine hypersensitivity. So I was thinking how this theory can be, or the paper or the finding can be utilized, in managing dentine hypersensitivity. And I think there was a question about it, if it's utilized already in the management or not. But to tell you the truth, meanwhile, still blocking the dentinal tubules, which is based on the hydromechanical theory, the previous one, still works and still blocks this cold sensation. But maybe, as my colleague here said, it may be other sensations.
Actually, it blocks the sensation to reach the receptors.
Yes, exactly. But now the elimination of the receptor or using another molecule that might block this receptor, yes, it might work. We will see what they will come up with. And thank you very much.
I would just add to what Monty is saying. The molecular basis of pathology is a hot thing. If you look at the last Nobel Prize, it was for immunotherapy for cancer. Then you're targeting expression of certain proteins and receptors which inhibit recognition of cancer cells. So everything is coming to that, and that is the way forward now for cancer, they are saying. So it applies to so many areas.
Yes, so this is the molecule that they put in the paper. So it has a strange name. It's called HC07 and HC030031. But that's, I don't know how healthy it would be. But that's the molecule that was the receptor. Yeah. So maybe it could be used for this molecule or similar molecules for adding to sensitivity.
Okay.
Sorry.
Okay, Prof. We have seen that they utilize very sophisticated methodologies, but you have already read the paper and many papers regarding this issue. Are there any limitations highlighted in the paper or from your point of view? In other words, how confident should we, when we're talking about this paper and results?
This laboratory evidence, so the ultimate thing is to have clinical trials and things like that. But in the context of biological evidence, these are very strong studies. Because they tested the hypothesis not with one experiment, but maybe with 20 different experiments to confirm that the hypothesis is correct. So they tested this way, and then that way, and then this other way. And when they found that all those experiments are pointing in the same direction, so probably what they are saying is correct. Yeah.
So my question was, those molecules, is their sole function is this one? Because I know that researchers are enthusiastic about blocking this action, but then with blocking it, are we not targeting other important function that this expression has?
Well, this molecule, this receptor in particular, the TRPA1, it has just been discovered quite recently, so the function of it is still not well known. So the way to understand well its function is basically that many molecular biologists they get a mouse that doesn't have it, and then they start analyzing everything in that mouse. Okay, so maybe these groups they looked at the teeth, but maybe there are other groups looking at the bones or at the brain or at the liver or whatever. So you start looking in everything in a mouse that doesn't have that molecule, and then you start understanding what is the role of that receptor in the animal. But that could take years, till they find all the factors.
Just a comment, Dr. Fadl. I think this research does refute the thermodynamic theory, because when they knocked out the receptor in these mice, the pain stopped. If the transmission of fluids contributed to the pain, you would have still found some of that through that other mechanism. So the only thing they knocked out on these mice was the receptor. So that might refute the theory, actually.
But did they not, because it wasn't typical, the mouse was not alive, right? It was like, it was a jaw. And then, so the other mechanism wasn't there. So when they tested this, the other mechanism did not exist.
This was in vitro.
And yes.
No, they have it in they're analyzing the electrical impulses in the nerve. So any pain sensation is coming through, they can detect it.
But the tubules are still there, and the mechanism could exist still.
Were they there?
Yeah, the tooth is there.
Yeah.
But that doesn't mean that blocking the dentines is a bad thing. What it means is that probably when you're blocking the dentines, what you're doing is you're protecting the receptor. Like the treatment that we are doing is effective, but the way it's working is different than what we thought it was.
Thank you. We have the network meeting starting in 10 minutes. I think only Professor Fale can break down a Nobel Prize paper and help the rest of us understand it. So thank you to him. And just one more reminder that the evaluation link for the CPD is posted in the chat box. So those who want it can access the link. And so thank you all for joining us today. And thank you especially to Professor Fale.
So how are you? I was in HMC this morning.